Welcome to the deep dive. This is where we tackle complex source material, kind of dense research papers and studies you might find in specialized labs, and we pull out the key insights, the really actionable stuff for you.
And today, yeah, we're diving deep into the microscopic but incredibly powerful world of advanced genome engineering.
Specifically, we're focusing on a tool that's become a real molecular workhorse, the PiggyBac or PB transposon system.
Right. And what's just amazing, really, is that this tool, which is now like transforming stem cell research and gene therapy, it actually comes from a pretty common garden pest,
No way. Which one?
the cabbage looper moth, Trachoplusianae.
That's incredible. A high-tech genetic tool from a moth munching on cabbage. Okay, so our mission today for you is to really unpack this piggyback system, how it works, why it's becoming so dominant in non-viral gene delivery, and especially how it gets super precise when you pair it with systems like Tedon for control.
Exactly. And we've got some great sources today, detailed research articles digging into the mechanics, its role in functional genomics, and even a cutting-edge study showing how it's used to make induced pluripotent stem cells, iPSCs, in black bone sheep.
Wow.
So by the end of this, you should have a really solid, nuanced grasp of where non-viral gene delivery is heading. The cutting edge.
Okay, let's start right at the beginning. This jumping gene idea, transposons. Can you just briefly define what a DNA transposon is?
Yeah.
And why are we using these things, which are mostly like dormant fossils in us, as clinical tools?
Sure. So DNA transposons are basically segments of DNA that can, well, move around the genome. They use a mechanism we call cut and paste.
Cut and paste, okay.
Yeah, they encode their own enzyme, a transposase, that does the cutting and pasting. Now, you're right, the ones native to humans or rodents are generally inactive, sort of fossilized. But scientists figured out how to take versions from other species, like PiggyBac from that moth, reactivate them, and then really refine them for gene transfer in mammals.
It shows how conserved these basic molecular machines are across life, doesn't it?
Absolutely, it's fundamental biology being repurposed.
Now, our sources immediately bring up a key comparison. Piggybac versus the other big name in non-viral transposons, Sleeping Beauty, or SB, which, funnily enough, comes from fish.
Right, salmonid fish, another unexpected source.
So if they both do this cut and paste thing, what's the really critical difference? Why is PB often getting the edge, especially for something sensitive like stem cell therapy?
It really comes down to safety and flexibility, and it hinges on what's left behind after the transposon moves, or rather, what's not left behind. SB was groundbreaking, don't get me wrong. First one really made to work well in human cells.
Okay.
But when SB cuts itself out of the genome excises, it leaves a little scar, a footprint, usually about 2 to 5 base pairs of altered DNA sequence at the original spot.
And that small scar, those few base pairs, why is that such a big problem clinically?
Well, think about gene correction. Let's say you're fixing a mutation in a patient's stem cells. You might need to insert a correct copy of the gene, maybe with a selection marker, let the cell use it, confirm the fix, and then you absolutely need to remove everything you added to show the cell is clean, right?
Completely clean.
Right. If removing the system leaves that footprint mutation, you haven't truly reversed the modification. You've left a permanent genetic alteration, a tiny scar, but it could still disrupt something important nearby, like a gene regulator.
Okay. So piggybac.
PiggyBac is different. It has this incredibly valuable feature, seamless excision. When the PB transposase cuts the transposon out, it does it perfectly. It restores the original DNA sequence exactly as it was before insertion, no trace left behind.
Wow. Footprint-free removal.
Precisely. And that is transformative for making clinically acceptable cells, like iPSCs, where you must remove the reprogramming factors you used to create them. You need that genetic slate wiped clean.
That seamless capability really is key. Okay, let's get into the mechanics. How does it actually do this? You mentioned it's not delivered as one piece. It's a binary system.
That's right. For safety and control, it's usually split into 2 parts, often on 2 separate plasmids. Think of it like a delivery truck and its cargo, kept separate until you need them.
Okay, truck and cargo.
So first, you have the cargo plasmid, the donor plasmid. This carries the gene you actually want to deliver your transgene, and it's flanked on both sides by specific DNA sequences called inverted terminal repeats, or ITRs. These are like the address labels the enzyme recognizes.
Got it. The ITRs mark the payload.
Exactly. Then on a second separate plasmid, the helper plasmid, you have the gene for the engine, the PB transposase enzyme, PBase.
So you introduce both plasmids into the cell. What happens then? How does the cut and paste work?
Right, so first, the cell needs to make the P-base enzyme from that helper plasmid. Usually this is done transiently, meaning the plasmid is designed so the enzyme is only made for a short time.
Okay.
The P-base enzyme then finds and binds to those ITR sequences on the donor plasmid, the ones flanking your gene. It makes the cut, cleanly snipping out the whole segment ITRs and the gene between them.
And then the pasting part.
Then the enzyme carrying this DNA fragment searches the cell's own genome, the host's genome, for a specific landing site. For PiggyBac, this site is a 4-base pair sequence, TTAA.
TTAA.
Correct. When it finds a TTAA-site, P-base makes cuts there and pastes the transposon sequence in, seamlessly integrates it. Now, these TTAA-sites are super common. There are hundreds of thousands across the genome, so insertion is still considered pretty random overall, but it does target these specific 4 bases.
Okay, that makes sense. Now, here's something that really jumped out from the sources. The sheer size of the cargo PB can handle, its cargo capacity. Why is this such a big deal compared to other non-viral methods and what needs such a huge delivery truck?
Yeah, the cargo capacity is maybe one of its most powerful features, especially for complex genetic engineering today. Older systems, including early SB versions, often really struggled once the cargo got bigger than, say, 5 or 6 kilobases.
6 kilobands, which limits you to basically one gene, maybe a small promoter.
Pretty much. But what if you need to deliver a whole set of genes, maybe multiple genes linked together with those self-cleaving 2A peptides we mentioned? Or what if you need to deliver a really large gene with its own complex regulatory elements, its enhancers and silencers, maybe spanning tens of kilobases? Or a massive protein coding sequence?
Like the dystrophin gene for Duchenne muscular dystrophy. That thing is famously huge.
Exactly. That's a classic example. Researchers have really pushed PB. They've successfully put transposons carrying DNA fragments up to 100 kilobases, that's massive, into mouse embryonic stem cells and shown they integrate with good integrity. Now, efficiency does drop off with size, naturally, but having that capability means PB can handle multiplex genetics. You can deliver not just one instruction, but a whole coordinated set of instructions, maybe an entire metabolic pathway or a complex regulatory circuit all in one go. It's the difference between sending a single text message and delivering a whole instruction manual.
That's a huge leap. Okay, so that's the basic machinery, but science always iterates, right? The sources talk about improving the P-base enzyme itself, making it work better than the original moth version in our cells. What were the key steps there?
Right. Optimization is key. The original moth enzyme wasn't perfectly adapted to the mammalian cell environment, so the first big step was making a version optimized for mice called MP-base. Codon optimization, basically making the gene sequence use the language mouse cells prefer, and that alone gave something like a 20-fold boost in activity compared to the original.
20-fold just from optimizing the code.
Yeah. Then came an enhanced version, ePiggyBac. This one used human codon optimization, and crucially, they also tweaked one of the ITR sequences it recognizes. That combination boosted integration efficiency another 10-fold, specifically in human embryonic stem cells.
Okay, better and better. But the real star seems to be the hyperactive version, high P-base.
That's the one generating a lot of excitement now, yes. High P-base. The performance jump was pretty dramatic. In standard lab tests, it outperformed the mouse version, MP-base, by about 10-fold.
10 times better again.
But more importantly, for actual therapeutic potential, they tested it in vivo for gene delivery to the liver, which is a tough target. And high P-base gave a 20-fold increase in gene expression there compared to MP-base in one key study.
Wow. How did they achieve that? Just more tweaking of the enzyme?
Essentially, yes. It usually involves making specific changes, point mutations, within the core catalytic part of the P-base enzyme, the bit that does the cutting and pasting the DDE motif. It's like fine-tuning the engine for maximum speed and efficiency within the specific environment of a human cell.
Okay, so now we have this super efficient hyperactive enzyme. But doesn't that create a safety problem? If it's so good at cutting and pasting, what stops the transposon, once it's nicely integrated, from just deciding to jump again later on? That sounds like genetic instability, exactly what you don't want.
That is the absolute key safety question. Secondary transposition. If the P-base enzyme hangs around too long, it could find the integrated transposon and move it somewhere else, potentially causing damage.
So how do they solve that paradox, make it powerful but controllable?
With some really clever molecular engineering. 2 main strategies. First, they tackled the enzyme's ability to reintegrate. They created excision competent but integration defective P-base mutants XMLs plus Ingem, meaning this modified P base can still recognize the ITRs and cut the transposon out cleanly, do the excision, but it has a defect, usually in that core catalytic region, that stops it from being able to paste it back in somewhere else.
So it's like a one-way removal tool. It can clean up, but it can't make a new mess.
Exactly. It allows for that seamless removal we talked about, but without the risk of it then reinserting randomly. Second, they tackled how long the enzyme itself sticks around. Delivering the P-base gene on a plasmid means the cell can keep making the enzyme for days or weeks.
Which increases the risk window.
Right. So the smarter approach now is often to deliver the P-base enzyme not as DNA, but as mRNA, messenger RNA.
Why mRNA?
Because mRNA is inherently unstable in the cell. It has a very short half-life. The cell naturally degrades it within hours. So if you deliver P-base as mRNA, the enzyme is only produced for a very brief period, does its job, either integration or excision, and then it's gone.
Much less time for it to cause off-target effects or secondary jumps.
Precisely. That transient expression drastically cuts down the potential for genetic toxicity. So you combine the XBA plus int mutant for removal with mRNA delivery for transient expression, and you have a much safer, highly controlled system. So we've got this great delivery system, PB, stable integration, high capacity, seamless removal. But that's only half the story for sophisticated engineering. The other half is control after integration.
Meaning you don't always want the gene you delivered to be on all the time.
Exactly. Often you need to be able to switch the gene on or off when you decide. Temporal control. And that's achieved by pairing the PB transposon delivery with what we call inducible gene expression systems.
Okay, inducible systems. Why go to that extra trouble? Adding chemicals or other triggers?
Right.
Wouldn't it be simpler just to have the gene stably expressed once it's in?
It seems simpler, but stable constitutive expression can actually muddy the waters scientifically. If you have a gene constantly on, especially if it's a really potent gene, maybe one involved in cell growth or, conversely, cell death, it can cause subtle but significant side effects over time. The cells might grow slower, or they might activate complex internal feedback loops to try and compensate for this constant artificial signal. It creates noise.
So you might not be seeing the direct effect of the gene you're interested in.
Exactly. If you want to know the immediate direct consequence of turning gene X on, you need to be able to flip that switch precisely when you're ready to observe. Inducible systems let you do that. They're reversible, they're flexible, and they let you isolate the primary effect, avoiding all those secondary long-term adaptations that can happen with irreversible changes like a gene knockout, especially in complex organisms.
Right, makes sense. Okay, so let's dive into the most popular inducible system out there, the tetracycline, or TET system. Everyone seems to use it, especially TET-on. What makes TET, and specifically its trigger molecule, doxycycline or DOX, the kind of go-to standard?
The TET system's success comes from a few things. It's based on bacterial antibiotic resistance genes, which are generally very safe and don't interfere with normal mammalian cell processes. Doxycycline, which is a stable and well-characterized analog of tetracycline, is the preferred inducer because, well, it works really well.
How so?
It has a very high affinity for the regulatory protein involved. It gets into tissues effectively. It has low toxicity, even at the doses needed for induction. And its biological half-life, around 24 hours, is well understood. And crucially, the TET-on configuration, which is the one most people use now, is known for having incredibly low leakiness.
Meaning the gene stays properly off when you don't add the DOX.
Exactly. Very little background expression, which is critical for getting clean results.
Okay, so how does TET-on actually work at the molecular level? What happens when you add the DOX?
TET-on relies on a specially engineered protein called the reverse tetracycline-controlled transactivator, or RTTA. It's a fusion protein.
A hybrid.
Yeah, it combines a part that binds DNA, specifically the TET operator or TET-O sequences, with a powerful activation domain, usually from the VP16 virus protein, which is great at recruiting the cell's machinery to turn genes on. Now, the reverse part is key. This RTTA protein is engineered so it only binds to the DNA target sequence in the presence of DOX.
So DOX is like the key.
Precisely. Without DOX, RTTA floats around, doesn't bind. You add DOX, the drug binds to RTTA, changes its shape, and now it can lock onto its target DNA sequence, which is called the TET response element, TRE. This TRE is placed right next to the gene you want to control.
Okay.
Once the RTTA-DOX complex binds the TRE, that VP16 part kicks into gear, recruits RNA polymerase and other factors, and powerfully switches on the expression of your target gene.
Simple on-off switch triggered by the drug.
Now, the sources mention advanced versions, like TET-on 3G systems, achieving just staggering levels of induction, like over 10,000-fold. How do they get that much better? Yeah, the fold induction can be massive with the newer systems. It comes from optimizing both components. The 3G systems use improved RTTA variants that bind docks even more tightly and are perhaps more stable. But the really big improvement was in the TRE promoter itself, the TRE3G promoter.
How do they optimize the promoter?
They carefully redesigned it to minimize binding sites for the cell's own natural transcription factors. See, sometimes endogenous proteins might weakly bind to an artificial promoter and cause a little bit of background expression, that leakiness. By removing those potential binding sites, the TRE3G promoter has an extremely low baseline activity when RTTA isn't bound.
So the off state is truly off.
Pretty much. Which means when you do add docks and the RTTA activates it, the difference between off and on the fold induction becomes huge.
Got it. And just to be clear, how does this compare to the older TED-off system?
PEDOF is just the opposite logic. It uses the original transactivator, TTA. This TKA protein is designed to bind the TRE and turn the gene on naturally in the absence of docks. When you add docks to a TED-off system, the drug binds to TTA and prevents it from binding to the TRE. So adding docks turns expression off.
So TED on, add docs, gene on N, TED off, add docs, gene OF.
You got it. TED-on is generally preferred now because of that lower leakiness in the off state.
Makes sense. Now, TED isn't the only game in town for chemical control. The sources mention others, like the QMate system and the tamoxifen system. How do they stack up?
They offer different flavors of control. The QMate system is quite similar to TED-on, actually. It uses QMate, another non-toxic chemical, as the inducer, and it can achieve similar high levels of induction, maybe 100 to 1,000-fold. Its main advantage is that it's orthogonal to TED.
Orthogonal meaning?
Meaning it works completely independently. You could have one gene controlled by TED-on docks and a different gene in the same cell controlled by QMate. They won't interfere with each other. That's really valuable for studying interactions between two pathways, for instance.
Ah, okay. Two separate switches. What about tamoxifen?
The tamoxifen system is different. It usually relies on fusing the Cre recombinase enzyme to a mutated estrogen receptor, ER, ligand-binding domain. This Cre-ER protein stays trapped in the cytoplasm until you add tamoxifen. Tamoxifen binding allows it to move into the nucleus and activate Cre's function, which is usually to cut DNA at specific LOXP sites.
So it's site-specific cutting, triggered by tamoxifen.
Right. But two key differences from TQMate. First, the induction level is generally lower, maybe 30 to 50-fold. Second, and most importantly, Cre-mediated recombination is irreversible. Once Cre cuts the DNA, that change is permanent. So it's great for things like permanently labeling cells for lineage tracing, but it doesn't give you that reversible on-off switching capability.
Right. Different tools for different jobs. Okay, now for the really futuristic stuff. Light switchable systems. Using blue light to turn genes on. How does that even work?
Yeah, this is super cool synthetic biology. These systems borrow light-sensitive proteins from other organisms, often plants or fungi. These proteins naturally change their shape or bind to other proteins when they absorb light of a specific wavelength, typically blue light.
Like photoreceptors in plants.
Exactly. Common examples are systems based on the VVD protein, which dimerizes, pairs up with itself, in blue light, or the CRY2-CIB1 system from Arabidopsis, where CRY2 binds to CIB1 only when exposed to blue light. Scientists have cleverly engineered these light interactions to control gene expression.
So instead of adding a chemical like dox, you just shine a blue light on the cells.
Pretty much. And the advantages are significant.
What are they? Faster? More precise?
Both. First, the kinetics are much faster than chemical induction. You can see gene expression start within minutes of turning the light on. It might peak in about an hour. And crucially, it shuts off almost immediately when you turn the light off. Much faster on-off cycles than waiting for a drug to diffuse in and then wash out.
Okay, speed is one. What else?
The other huge advantage is spatial control. With a chemical like dox, you add it to the whole dish or inject it into the whole animal. All the cells with the system get induced. But with light, you can use a focused beam, like from a microscope or an LED array.
Meaning you could turn the gene on in just one cell, or even part of a cell.
Precisely. You can illuminate just a specific group of cells in a tissue, or even target a subcellular region, like just one end of a neuron. That level of spatial precision is impossible with chemicals. It opens up amazing possibilities for studying cell-to-cell communication, development, or maybe even super-targeted gene therapy in the future.
That's incredible. So maybe lower overall induction levels than the best TET systems, the sources said, maybe 50, 100-fold, but unparalleled speed and spatial targeting?
Exactly. It's a different kind of precision.
So we have PB for delivery stable, large cargo, seamless removal,
Mm.
and we have these sophisticated inducible systems, TET, QMate, light for control.
Yeah.
How do these partner up with the other major genome editing tool, CRISPR-Cas9?
They work together beautifully, especially in gene correction scenarios. Let's go back to fixing sickle cell disease in patient-derived iPSCs. You use CRISPR-Cas9 to make the specific cut at the faulty gene. Then you need to provide the correct DNA sequence as a template for repair. Often you also include a selectable marker gene temporarily, just to easily identify the cells where the correction actually happened.
Okay.
Now, once you've selected the corrected cells, you don't want that extra marker gene hanging around permanently. That's where PB shines again. If that marker gene was delivered flanked by PB's ITR sequences, you can bring in the P base enzyme transiently, maybe as mRNA, and it will seamlessly excise just the marker cassette, leaving behind only the perfectly corrected patient gene.
So PB acts as the cleanup crew after CRISPR does the precise surgery.
Exactly. It facilitates the traceless removal of the temporary components needed for the editing process. It's a really elegant synergy.
One more point on precision. Can we make PD itself insert less randomly, instead of just hitting any old TTAA site?
People are definitely working on that. It's more experimental, but the concept involves fusing the P-base enzyme directly to a component of the CRISPR system, specifically a dead Cas9 or dCas9.
A dead Cas9.
Yeah, dCas9 is a version of the Cas9 enzyme that's been mutated so it can no longer cut DNA, but it can still be guided by a guide RNA to bind to a very specific target sequence in the genome. So if you create a fusion protein, dCas9 P-base, you can use the guide RNA to bring the P-base enzyme directly to a chosen spot in the genome.
So you're essentially telling P-base exactly where to paste its cargo.
That's the idea. To target PB transposition to specific safe harbor locations in the genome rather than relying on random TTAA integration, it's still under development, but it holds huge promise for improving the safety and predictability of PB for future clinical use.
Okay, so we have this incredibly versatile and controllable PB toolbox. Let's shift gears now and look at how researchers are using it, particularly in functional genomics, basically, figuring out what different genes actually do. A big part of that is genetic screening.
Right, forward genetic screens. The idea is you create a huge library of cells, each with a different random genetic mutation, and then you apply some kind of pressure or test, maybe expose them to a drug, or change their growth conditions and see which mutations cause a specific outcome or phenotype. That tells you which genes are important for that process.
Sounds powerful, but the sources highlight a major historical challenge doing this effectively in mammalian cells. What was the big hurdle?
The hurdle was diploidy. Most mammalian cells have 2 copies of almost every gene, one from each parent. Now, many mutations are recessive, meaning you only see an effect if both copies of the gene are inactivated.
So you need to hit the same gene twice in the same cell to get a clear loss-of-function signal.
Exactly. And generating homozygous knockouts, inactivating both copies, randomly and efficiently across the whole genome in a diploid cell population is technically very difficult and time-consuming. It really limited the scale and power of forward screens in mammals compared to, say, yeast, which can easily exist as haploids.
Yeast are simpler. So what was the breakthrough solution for mammalian cells?
The breakthrough was the development and refinement of haploid embryonic stem cells, or HESCs.
Haploid stem cells, meaning they only have one set of chromosomes.
That's right, just one copy of each gene. And suddenly, that whole problem of recessivity disappears. If you make a single mutation that knocks out a gene in a haploid cell, you immediately see the resulting phenotype. It behaves like a dominant mutation.
So PB-mediated gene disruption becomes way more effective in these haploid cells.
Absolutely perfect match. PB allows you to rapidly and efficiently create huge libraries of these haploid ESCs, where each cell has potentially one gene knocked out by a random PB insertion. You can generate genome-wide mutant libraries relatively easily.
And then screen those libraries for interesting phenotypes.
Precisely. It brings the power and throughput of yeast genetics to mammalian stem cell biology.
Okay, let's look at some concrete examples from the sources that use these PB-HSC screens. What did they find regarding drug resistance, for instance?
They proved really useful there. For example, researchers screened for resistance to the drug olaparib. This is a PARP inhibitor used in cancer therapy. The PBAC screen quickly and cleanly pinpointed the gene PARP1 itself. If you knock out PARP1, the cell becomes resistant to the drug.
So it validated the drug's known target very efficiently.
Exactly. It showed the platform worked. In another study, they screened for sensitivity to doxorubicin, a common chemotherapy agent that damages DNA. The screen identified several genes, including RMI2, PDK4, and XEBD6, whose loss made the cells more sensitive to the drug. That kind of information is vital for understanding chemoresistance mechanisms.
Very powerful for pharmacology. What about using it to understand basic stem cell biology?
Yeah. They've used it to probe fundamental processes. One screen looked for genes required for stem cells to properly differentiate or exit from self-renewal. That's a critical step. The PBAC screen identified several key regulators, including an RNA-binding protein called PUM1 and a zinc finger protein, ZFP706.
What happened when those genes are knocked out?
The cells basically got stuck. They had trouble exiting the self-renewing state and committing to differentiation. It showed these genes are crucial gatekeepers for that process.
That's fascinating. There was also a really clever finding about the haploid cells themselves, wasn't there? They tend to spontaneously become diploid, which is a problem for experiments.
Yeah.
Did PD help figure that out?
Yes, that was a really neat application of the technology. They essentially screened the PBAC library to find mutations that help the cells stay haploid for longer.
A screen for stability itself.
Right. And they found that knocking out a gene called EDL4 significantly reduced the rate at which the haploid cells spontaneously diploidized. But the really cool part was why. They linked EDA4 function to a switch in energy metabolism.
How so?
They found that EDA4 deficiency, which stabilized haploidy, was associated with a shift away from glycolysis towards oxidative phosphorylation. It suggested a link between the cell's energy state and its ability to maintain that single chromosome set. It offered a completely new metabolic strategy for keeping these valuable haploid cells stable in culture.
Wow. Using the tool to improve the tool itself. Okay, so PB is great for these loss-of-function LOF screens where you knock genes out. Can you also use it for the opposite gain-of-function, GOF screens, where you're maybe overexpressing genes?
Absolutely. You just designed the PB transposon differently. Instead of just being an insertion that disrupts a gene for GOF screens, the transposon itself carries strong on signals, typically powerful viral promoters or enhancers like from the CMV virus.
Okay.
When this GOF cassette inserts randomly into the genome, it can land near a gene and force its massive overexpression. Or the transposon might carry a truncated version of a protein that acts in a dominant way, or it might just generally activate transcription in the region where it lands. The goal is to see which activation events lead to a phenotype of interest.
So looking for genes that cause a change when turned up too high. What's a good example of a finding from a PBGOF screen in human cells?
There was a nice example screening human pluripotent stem cells, iPSCs. They were looking for genes that conferred resistance to a specific drug called plurisin-1, which is toxic to stem cells. The PBGOF screen found that overexpression of components of the RAS pathway, a really well-known signaling pathway, often hyperactive in cancer, made the iPSCs resistant to the drug.
Directly linking RAS activation to resistance.
Right. And in the same screen, looking for factors that help the cells grow better in culture, they found that activating genes that inactivated a different pathway, the RHO-ROC pathway, gave the cells a significant growth advantage. So these GOF screens can rapidly map pathways involved in drug response, survival, or growth.
Okay, so building on those functional studies, PB, especially with its safety features, has become really central for generating more specialized stem cell lines, the kind needed for preclinical testing and moving towards actual therapies. Let's start with the big one. Induced pluripotent stem cells, iPSCs. We touched on this, but the original methods using viruses to reprogram normal cells back to stem cells, they worked, but left behind viral DNA, right? Big safety concern about cancer risk. How did PB change the game there?
PB offered a non-viral and crucially excisable solution. Researchers quickly figured out they could put all the necessary reprogramming factors, the famous Yamanaka factors, OCT4, SOX2, KLF4, CMOK, maybe others, onto a single large PB transposon vector. Remember that high cargo capacity?
Right, put them all in one package?
Yeah, often linked by those 2A self-cleaving peptides, so they're expressed as individual proteins from one transcript. They'd use this PD vector to deliver the factors, reprogram the somatic cells into iPSCs. And then, here's the key part. Once reprogramming was successful and the cells were stable iPSCs, they could bring back the P-base enzyme transiently.
And cut the whole factor cassette out.
It's
Yeah.
exactly, seamlessly excised, leaving behind an iPSC line that was generated without any residual foreign reprogramming DNA integrated into its genome. This massively improved the safety profile and made iPSCs much more viable for potential clinical use. No viral baggage, no permanent transgene integration from the reprogramming process itself.
That's huge for clinical translation. So with that improved safety, how is PB being used now to actually repair genetic diseases in patient cells?
It's playing a critical role in gene correction therapies, often teamed up with CRISPR or other editors like TALENs. We mentioned sickle cell disease, SCD, and beta thalassemia. For these diseases, scientists take cells from a patient, reprogram them into iPSCs using maybe a temporary method, then use CRISPR to fix the specific gene mutation. But the editing process often requires, again, temporary helper elements, like a marker gene or a repair template delivered on a plasmid. PB is frequently used as the vehicle for these temporary elements because, after the CRISPR correction is done and verified, the PB system allows for that clean, footprint-free removal of all the helper DNA. The end product is the patient's own iPSC line, now genetically corrected, with no leftover vector sequences.
The ultimate personalized cell therapy platform made safer by PB. I also saw a mention in the sources about using PB for disease modeling, specifically for SARS-CoV-2.
Yeah, that's a good example of its versatility beyond just inherited diseases.
Right.
Researchers created a human embryonic stem cell line using PB combined with the TETON system. This line was engineered to express the SARS-CoV-2 nucleocapsid N protein, but only when doxycycline is added.
So they can switch on a key viral protein in human stem cells on demand.
Right. It creates a controlled model system. They can turn on the N protein expression at specific times during differentiation, for instance, and study exactly what pathological effects it has on different human cell types derived from those ESCs. It's a powerful platform for dissecting COVID-19 mechanisms and potentially screening for drugs that counter the N protein's effects.
Very clever. Okay, beyond iPSCs and ESCs, what about other important stem cell types? Hematopoietic stem cells, HSCs, the blood-forming stem cells.
PB is definitely being used for stable gene transfer into HSCs, yes. Again, often for diseases like SCD, the goal is to deliver a correct copy of the beta-globin gene into the patient's HSCs, which can then hopefully repopulate their blood system with healthy red blood cells. Studies have shown PB-modified HSCs can lead to a reduced sickle phenotype in mouse models.
So it works there too. Is it the best option?
Well, it's effective, but the sources note that in HSC specifically, PB faces stiff competition from the hyperactive sleeping beauty transposase, SB100X, which also shows really high activity in that particular cell type. So for HSC gene therapy, it's still a bit of a horse race to see which non-viral system ultimately proves the most efficient and safest long-term.
Interesting. What about mesenchymal stem cells, or MSCs? They're used a lot in regenerative medicine and as potential delivery vehicles.
PB works very well for gene integration into MSCs. They're generally easier to transfect than HSCs. And the sources highlight a really cool potential therapeutic application using PB-modified MSCs for cancer.
How so?
Researchers engineered MSCs derived from fat tissue using PB to stably express interferon gamma, IFN gamma, a potent immune-stimulating molecule. What was fascinating was that these engineered MSCs seemed to naturally home in on the tumor microenvironment, specifically the supportive tissue or stroma around the tumor, in a mouse model of melanoma.
So the MSCs acted like delivery trucks, carrying IFN gamma straight to the tumor.
Exactly. By delivering IFN gamma locally, right where it's needed, they could inhibit tumor growth and blood vessel formation, angiogenesis, much more effectively and potentially with fewer systemic side effects than just injecting IFN gamma everywhere. It shows PB can help create these smart, targeted cell therapies.
That's a very targeted strategy. And finally, muscle disorders. We mentioned Duchenne muscular dystrophy, DMD, and the huge dystrophin gene.
Right. For DMD, PB's high cargo capacity is a major asset. Researchers have successfully used PV to deliver therapeutic dystrophin genes into myoblasts, which are the muscle precursor
A
cells.
full-length gene, or smaller versions.
Both have been explored. Delivering the massive full-length dystrophin is challenging for any vector, but PB can handle it better than many others. More commonly, they use PB to deliver highly functional but shorter microdystrophin genes, which are still quite large but more manageable. The idea is you modify the patient's myoblasts with PB carrying the microdystrophin gene, and then these cells can fuse and repair muscle tissue, providing a lasting source of the missing protein.
So PB offers a potential non-viral route for DMD gene therapy. And just quickly, CAR T-cell therapy for cancer.
Yes, PB is definitely entering the CAR T space as well. Building better CAR T cells involves getting stable, high-level expression of the chimeric antigen receptor, the GRR, that targets the T cell to the cancer. PB is being explored as a non-viral way to integrate that CR gene reliably into the T cells, potentially offering safety or manufacturing advantages over the traditional viral methods used for CAR T production. Okay, so to really tie all these threads together, the PB system, the TET-on control, the application in stem cells, let's look at this detailed case study from the sources involving the black bone sheep. It's a great example of putting the whole system through its paces in a large animal model.
Black bone sheep. Why that particular animal? And what exactly were they trying to achieve by reprogramming their cells?
Well, the black bone sheep is an agriculturally important species, and large animals like sheep are often used in preclinical research because their physiology is, a bit closer to humans than mice are, especially for things like regenerative medicine or cloning technology. The specific goal here was to take ordinary skin cells, fibroblasts, SFs, from these sheep, and reprogram them into high-quality induced pluripotent stem cells, iPSCs.
Using the PB system.
Exactly. They used the combined PiggyBac plus TET-on transposon system. The PB part delivered the reprogramming genes, and the TET-on part allowed them to switch those genes on using doxycycline only when needed.
Which reprogramming genes did they use? The standard 4.
They actually used a cocktail of 7 factors. The standard ones OCT4, SOX2, KLF4, macaque, using the bovine versions, but they also added porcine nanog and Lin28, plus SV40 large T antigen, which can help with efficiency and cell survival during the stressful reprogramming process.
7 factors, all delivered by PB and controlled by TET-on. That's complex. How did they know they'd actually made good iPSCs, suitable for use in a large animal?
They did a lot of validation checks. First, the morphology. The reprogrammed cells formed nice, compact colonies typical of pluripotent stem cells. They stained positive for alkaline phosphatase, another classic stem cell marker.
Okay.
Crucially, they checked the chromosomes. The iPSC clones showed remarkable genomic stability. Even after being grown in culture for many passages, about 90% of the cells maintained a normal, stable sheep karyotype, which is 2n equals 54. This suggested the PB integration and reprogramming process hadn't caused major genetic chaos.
That's reassuring.
And finally, they confirmed that key pluripotency proteins, OCT4, SOX2, NANOG, were being expressed and located correctly within the nucleus of the iPSCs, confirming their undifferentiated stem-like state.
Okay, so they look like iPSCs, they had the right markers, stable chromosomes, but the sources went deeper, using transcriptome sequencing to compare the iPSCs to the original fibroblasts. What did that reveal about why these iPSCs might be better or different?
That's where it got really interesting. The sequencing showed a fundamental shift in the cell's core programming, especially around energy and survival. Compared to the starting fibroblasts, the newly generated iPSCs had significantly higher activity in 2 key pathways. Oxidative phosphorylation, which is the main way cells efficiently generate energy, and cell proliferation signaling pathways.
So the reprogramming didn't just flip the pluripotency switch, it sort of supercharged the cell's engine and growth signals.
That's a good way to put it. It seemed to rewire their basic metabolic and survival strategies.
Can you give some specific gene examples of that shift?
Sure. They saw clear upregulation of genes involved directly in oxidative phosphorylation, like ATP5B and SDHB. At the same time, genes that push the cell cycle forward, like CDK1, cyclin D1, and genes that prevent programmed cell death, or apoptosis, like the key anti-apoptotic gene BCL2, were also significantly upregulated.
And what about genes that promote cell death?
Conversely, a major pro-apoptotic gene, BAX, was found to be significantly downregulated in the iPSCs compared to the fibroblasts. So the whole picture was one of cells that were metabolically active, highly proliferative, and strongly resistant to dying. Essential traits for stem cells that need to build tissues.
Okay, so they had these high-quality, robust iPSCs. What was the ultimate test of their actual developmental potential in this large animal context?
The ultimate test was somatic cell nuclear transfer, or SCNT, basically cloning. They took the nucleus out of a sheep egg cell. Actually, they used readily available bovine oocytes as recipients here, and replaced it with a nucleus from either one of the new iPSCs or, as a control, one of the original fibroblasts. Then they saw how well these reconstructed embryos developed.
And what were the results of that comparison?
The results were striking and really validated the quality of the iPSCs made with the PB-TET system. The embryos reconstructed using iPSC nuclei had a significantly higher cleavage rate, meaning they successfully started dividing about 85%, And even more dramatically, they had a much higher rate of developing to the blastocyst stage, which is a critical early embryonic stage, around 52%.
How did that compare to using the original fibroblasts?
Much lower. The fibroblast-derived embryos had significantly lower rates for both cleavage and blastocyst formation. It clearly showed the iPSCs possessed superior developmental potential.
Why such a big difference? The sources suggested it might be related to how well the donor nucleus syncs up with the egg cell.
Yes, that's thought to be a major factor. The iPSCs, being undifferentiated and actively dividing, tend to be mostly in the G1 phase of the cell cycle. The recipient egg cell, after its nucleus is removed, nucleation, is typically arrested in the M phase, metaphase. Having a G1 donor nucleus sync up with an M-phase cytoplasm seems to work much better for proper chromosome replication and avoiding errors during those first critical divisions.
Whereas the fibroblasts might be in different cell cycle stages, leading to problems.
Exactly. Differentiated cells like fibroblasts are often quiescent, G0, or scattered throughout the cell cycle, leading to poorer synchronization and more developmental failures than SCNT. Plus, remember that metabolic rewiring.
The oxidative phosphorylation boost.
Right. The iPSCs, with their enhanced energy production capacity, are likely just better equipped to handle the massive energy demands of kickstarting embryonic development compared to the more metabolically constrained fibroblasts.
And did they also confirm these iPSCs could actually form different tissue types?
They did. As a final check, they showed that the iPSCs, when prompted to differentiate in vitro, significantly increased the expression of marker genes representing all three primary embryonic germ layers: DCN and FOXA2 for endoderm, KDR and CD34 for mesoderm, and Neurod for ectoderm. This confirmed their triploblastic potential, the ability to give rise to all the major tissue types in the body.
So a really comprehensive demonstration in a large animal model. PB plus TEDON delivered functional, stable, developmentally potent iPSCs. But safety. Even with seamless excision, what are the lingering concerns when thinking about using PB clinically?
The main one, as we touched on, is still insertional mutagenesis. Even though PB cuts itself out cleanly, the initial insertion is still largely random, happening at one of those many TTAA sites. And the sources note PB seems to have a slight preference for integrating within active transcriptional units, basically, within genes that are currently switched on.
Which raises the worry that it might land in the middle of an important gene and disrupt it, or land near an oncogene and activate it.
Exactly. Disrupting a tumor suppressor gene or activating an oncogene is the classic concern with any integrating vector. Now, mitigations like using mRNA delivery for the P-base help limit ongoing risks, and important long-term safety studies in mice using PB-modified cells didn't show any obvious tumor formation over a year, which is good news.
But still things to watch.
Definitely. Two other potential, though perhaps lower frequency issues are sometimes mentioned. One is the accidental integration of bits of the plasmid backbone DNA along with the transposon itself if the cutting isn't perfectly clean. The other is the possibility that the ITR sequences themselves, particularly the 5' ITR, might have some weak unwanted promoter or enhancer activity on their own, potentially affecting nearby genes. These are things that need very careful checking and vector design optimization before widespread clinical use.
Right, constant vigilance needed. Okay, we've covered a huge amount today, from moths in the garden to potentially fixing human genetic diseases and improving livestock breeding.
We really have. And I think what comes through is how PiggyBac, especially when you pair it with sophisticated control systems like TEDON or maybe even light switches, moves way beyond just sticking a gene in somewhere. It offers this powerful combination: high cargo capacity, that crucial footprint-free removal, and increasingly precise control over when and where the genetic modification is active.
Yeah, what really stands out for me is that increasing level of precision. The idea of using light for spatial control is just mind-blowing. And the cleverness of using PB and haploid cells to suddenly make recessive genetics easy to screen, that's revolutionary for basic science. And the black-bone sheep study, that was just a fantastic concrete example showing these tools aren't just theoretical. They're building a really solid foundation for using stem cells in large animal models, which is often that vital step towards human therapies.
Which brings us to a final, perhaps provocative thought for you, our listener. We've seen PB, often with TEDON, enabling high-precision reversible genetic changes in valuable stem cells like iPSCs. We've seen these non-virally generated iPSCs show superior developmental potential compared to older methods or starting cells. So the question is, How quickly will this kind of advanced, non-viral, footprint-free technology like PiggyBac actually replace the older integrating viral vectors as the definitive gold standard for generating cells for clinical-level gene and cell therapy applications in humans? Is that shift already underway?
That really is the multi-billion-dollar question for the future of genetic medicine, something to definitely keep an eye on. Thank you for joining us for this deep dive. We hope you feel thoroughly informed.